Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes we have talked about transcriptome read mapping and then we talked about transcriptome assembly. We started discussing quantification of transcript abundance and this is a very important step, and in this class we will be continuing discussion on this transcript abundance quantification. Okay. So, here are the concepts that we will be covering in this class. So, we will talk about read data to count data process, we will talk about some tools and we will also have a look how to do this ourselves with a small hands on, and then we will talk about alignment free approaches for transcriptome quantification. Right. So, many of the tools that we have discussed, they rely on alignment map or SAM or BAM files for this quantification, and some of the tools that we will discuss, they also rely on these alignment maps. But there are tools that actually can do this without doing any alignment, and they are actually significantly faster than alignment based tools. So, we will discuss those as well. Here are the keywords: raw count data and model based estimation. Okay, so, here is the data processing pipeline again for RNA sequencing data. We have talked about mapping, we have also talked about assembly in very brief, and we have come to the quantification step. Now, what we have discussed, some of the tools that can do assembly, they can also do quantification. For example, we have talked about string tie, you have talked about cufflinks, so they can also do quantification after the assembly process. In this class, we will be talking about some of the tools that actually are dedicated for the quantification purpose, because this is a very important process for any downstream analysis, right. So, we want this accurate, very accurate quantification of the transcript abundance for any inference later on. Okay. So, if you make mistakes here, those will lead to erroneous inferences. Okay. So, this is a very important step. So, this is something we will discuss today, quantification. So, there are several tools. Cufflinks we have discussed already, but then you have something called HTSEC count, feature counts, RSM, etc., and we will discuss those in this class. Okay. So, HTSEC count, it actually simply counts the number of reads that map to each gene, transcript or feature, right. So, this is the simplest of all the tools. It takes the alignment map and then the feature file, and it will simply count how many reads map to each gene, transcript or feature. Okay. So, these are the input files, right, the alignment map in SAM format, and you can give feature annotation, which is a GFF file or GTF file, and then output what you will get is a table with counts for all features. Okay. So, this is a very simple process, we just simply count. And what are the challenges here? Of course, this is a very simple process. So, one is if you have reads that map to multiple features or multiple genes. Okay. So, this is something that again we go back to the mapping to the repetitive sequences, right. So, if you have reads that mapping to multiple features and genes, how do you count those reads, right? Do you count them for both genes or both features, or do you just discard them, right? So, what are you going to do? And this is something that is a very difficult question to answer, right, and we cannot answer this by simply by a simple counting process. Of course, there are more sophisticated models that can deal with these kind of reads that actually map to multiple features or multiple genes. There is another tool which is called feature counts, which is very similar to the HTSEC count. So, it provides gene level counts and it will not provide any count for isoforms, right. Again, as we have seen, doing this for isoforms is very difficult, right, identification of isoforms, especially from short read data. So, again here the input files are alignment map, SAM BAM file, and then you have the feature annotation file, which is the GFF file. Okay. And here is the package which you can use, download and use, or you can get from R in Bioconductor and you can use this, right. So, again, if you are not familiar with R, you can simply go with the subread package and download and use that package. Okay. So, what we are going to do is we will have a hands on now, we will just simply look at this raw RNA-seq data and we will get to the raw count data, and using this very simple step Okay. We will not talk about the QC and pre-processing because you already know this, right, first QC etc. If you need to do trimming etc., you can do that. What we are going to do is we will take an example of this raw RNA-seq data, we will map using HiSET2, of course, only a small part of it, and then we will try to quantify with HT-sec count. Okay. It is a very simple step that I can show you right here right now. Okay. Okay. So, let us now go to the terminal, okay, and we will open the terminal, okay, and here we are, and what we need are these two tools. So, one is the HiSET2, right, and another one we need is the HT-sec count. Okay. So, here is the HiSET2 tool, this is the link where you can find this, okay, and here is the link of HiSET2 where you can find the tool, and again you can check the manual here. Okay, here is the manual, right, and you can find a lot of details, okay, and how to download etc. install. In addition, how to actually run the HiSET2, what are the options etc. Okay, so what I have done is I have actually installed this tool already and in my system, and so I can simply use this. Okay. So, you can see here, right, we have this HiSET2 folder, I have this HiSET2 folder here that I have actually. So, this is the program that I downloaded, this is the compressed file, then I have extracted and then I have actually added this to the path. So, I can simply call HiSET2 and I can get all the options etc. Okay. So, this is what I am going to do, right. I am just going to run this HiSET2 minus minus help. So, it will tell me what are the options etc. Okay. So, again what you will see is like HiSET2 is very similar to Bot-I2 because again it uses this BWT algorithm, right, the Barrows-Wheeler transform. So, the tool has also been kept very similar to use. So, it is actually very easy to use if you know this Bot-I2, right. So, we have HiSET2, we have minus x, again we have to give the index, then we have the minus 1 m1 minus 2 m2 minus ur, right. So, we are going to run this right here now. Okay. All right. So, all these options are there. So, you can simply run this with a data set. Now, what I have done is I have downloaded one data set here from NCBI GEO, okay, and I will show you that later during the hands on, when we have a full hands on how to download these data sets. And from this FASTQ I have created only a part of that file, right. I have just created a small file from that because that file is quite big, it will take a long time to run or to map. Okay. So, if you want to do this in a short period of time, we will just use this part file T1 part dot FASTQ. Okay. So, let us run this now. I said 2 minus x, right. Now, we need the indexes. Okay. Now, what is good about this is that HiSET2 can build these indexes, but you can also download indexes for some genomes. Okay. So, for example, you can check here, you have for specific species here, you already have these indexes built. Okay. So, you have this Homo sapiens etc. So, I have downloaded this one of the index which is for Saccharomyces cerevisiae, because the data that I have downloaded is for this species Saccharomyces cerevisiae. Okay. And you can see different types of indexes, right. So, you have genomes SNP, right. So, you have these single nucleotide polymorphisms with the genome that has been added, genome transcriptome, right. So, you have genome SNP's transcriptome, etc., right. And similarly, for different species you will see these different types of indexes. Okay. And for Saccharomyces cerevisiae you have genome as well as genome transcriptome. So, I have downloaded this genome transcriptome. If you just click on this, okay, so it will download, it will take a lot of time. So, I will just stop. I have actually downloaded this already and it is present in here. So, if you check, this is the R64 underscore tran, this is the sequence, this is the index file that is present in this folder. Okay. So, we will use this index. Okay. Okay. So, we just need to give the base name, right. We do not need to give all those dot 1 dot ht2 dot 2 dot ht2, right. This is very similar to both I2, that remember we just use the base name for this purpose. Okay. So, the next step is we have to give minus u, right, because we are working with unpaired data, we do not have paired end data, right, and this is the T1 part dot FASTQ, this is the FASTQ file that we want to analyze, and then we have minus s, right. We can simply say, right, part isat out dot SAM, right. Again this program gives output in SAM format, right. So, we will have to again mention minus s part is isat out dot SAM. Okay. So, part means this is only part of the full data we are working with. So, that is why I gave this name part. Okay. Okay, maybe we can just say T1 part, right, because this is from the T1 part file. All right. So, let us run this and see. Okay. So, what is the output? We have 25,000 reads. Okay. These are unpaired reads. Then it gives very similar statistics like both I2. Okay. You can see this, right. So, some reads did not align, some reads aligned exactly one time, and some reads about 7.5% reads aligned multiple times. Now, this is where this part is actually interesting, right. This is what we will do with these reads. Okay. If they are aligning to multiple genes, whether you count for both genes or you just simply discard from your analysis. Okay. So, this is where more sophisticated tools actually are important, right. So, which we will discuss a bit later, right. What are these tools that we can use to kind of use these read data that align to multiple positions in the genome or in the transcriptome, and how do you utilize those to actually estimate transcript abundance. Okay. Because these are discarding them means we are underestimating maybe the abundance of some transcripts, right, and counting them for multiple genes or transcripts might lead to overestimation for some abundance of some transcripts. Okay. So, this is a very important problem, right. It is not just for genome, not like just genome sequencing. Here the count, the number of reads mapping to each feature is very important. So, the overall alignment rate is 95.5%. So, this is a good alignment rate. So, we can now go ahead with the quantification. Okay. All right. So, let us see now if we can do the HT-sec count, right. So, let us see if we can. Okay. So, I have also opened this. So, here is the function HT-sec count, right. So, how do you actually use this function? Okay. So, again here you have all the details of these, right. You can find a lot of these options, and you have to install a program called HT-sec. You can see this, right, this program, and again the prerequisites and installation, these are all given. Okay, and you can check them and you can see that how do you install all this, right. So, if you are using Python, you can use this pip install HT-sec, or if you are in Linux, even Ubuntu, you can simply say sudo apt-get install, right. We have talked about this in Linux installation process, right. We can do that, right. Simply we can use this sudo apt-get install, and then of course, in Windows you can also have different dialog packages, etc. So, let us see if we can run this, right, HT-sec count. Here are the options. Again, we will not use any options. So, what it needs is the alignment file, the SAM file that we generated, and the feature file, right, the GFF file. Okay. So, we have downloaded the GFF file. So, let us see, right, the GFF file is here, GFF GTF. You can see this, right, inside this folder, right, and I have simply just copied this GCF 64 genomic news.gtf. This is the file that you can simply use. Okay. So, let us see if we can run this HT-sec count, because I have already installed this, right. So, what it needs is the alignment file, which is the SAM file. Okay. And this is the SAM file that we generated. Okay. And we need the GFF file, so or GTF file. So, I am using this .gtf, and let us see. Okay. So, it should run and it will generate statistics. okay, and you can see these statistics. Okay. So, what I am going to do is I am going to save this into another file. Okay. And inside a text file. Okay. So, you can see, right, so that we are not getting the output in the terminal, right. I can, what I can do is simply run it again, so that you can see clearly. Okay. And it gives out some statistics, etc., right. And what you want to do is we want to check this count file, right, that we generated, okay, because this will contain the data. Okay. And this is where you can see the raw count now. Okay. So, the first column you see is the gene names, okay, and the second column is the raw count data. Okay. What it simply says, okay, there are, for example, in this gene, right, you have 9 reads mapping to this gene. For this read you have, for this gene you have 32 reads mapping to this gene, right. Similarly, you can see this, right, for many genes you have these kind of statistics. Okay. Now, you might say for many genes we see 0, right, that is expected because we are working with only part data. Okay. We are not going working with the full data, right. If we work with full data, we can see counts for all genes almost. Okay. So, what I have done, actually I have processed the full data with HiSet2 before, and I have created this file which actually is present here. You can see this HiSet map out.sam. Okay. And this is the processing the full FASTQ file here. Okay. It took some time, right, few hours. So, that is why I had to run in the background, and then I have generated this SAM file, so that I can show you how the full output would look like. Okay. And then I have processed this with the htsec count, and you can see this raw count HiSet map out.txt, right. So, let us have a look at that raw count file, because this is after processing the full data, okay, not just the part data. And you can now see for most genes you have some counts, and for some genes the counts are very high, right. For example, you can see this gene here, right, you have 48,000 reads mapping to this gene. Okay. But still you have some genes that are probably not expressed because you see this 0 count, right. And now, because you have so much data, you can be sure, okay, these genes are probably not expressed or have very low expression. But what you see for most genes you have expression, right. You can see some read counts, and this is the raw read count. Okay. And this is a very important step for all subsequent data analysis. Okay. So, hopefully this is clear how we actually simply generate this kind of raw count data from the mapping, okay, using a tool like htsec count. Okay. So, what we will do is now we will talk about some of the more sophisticated tools that actually can deal with these multiple mappings, etc. Okay. So, let us go back to the presentation and let us continue our discussion on some of the other tools that are available and that can deal with this multiple read mapping. Okay. So, one such tool is called the RSEM. Okay. So, this tool is, the full form is RNA sequencing by expectation maximization. Okay. And so, this tool does not require any reference sequence, it can assemble transcripts as well as quantify transcripts. Okay. So, we have discussed like many such tools before. Now, there are several steps here, right. So, one is generation of reference transcripts from the read data, right, and then the reads are aligned against the reference, okay, and based on this alignment then you have the estimation of transcript abundance. Okay. So, what is unique about this tool is that it generates something called maximum likelihood estimate of abundance along with confidence intervals. Okay. So, it kind of gives you, okay, what is the 95% confidence interval for each feature, right. So, it is not just a single number for count, you have also a confidence interval, right. So, if that confidence interval, if that value is like if the variance is very high, right, then you probably cannot trust the data that you are getting. Okay. And a second unique feature is that it utilizes something called expectation maximization algorithm. Again, we are not going into details of this. So, it is again related to maximum likelihood estimate, but it goes through two steps. One is the expectation or E step, and then you have the M step. Okay. So, it performs maximum likelihood estimation. So, then it actually does this in an iterative process. Okay. So, you have E step, you have M step, then again E step until you converge. Okay. Again, we are not going into the details of this, but again you are encouraged to actually look into these algorithms if you are also interested in the algorithm part. So, what we get after this tool is that we get gene and isoform level estimates of abundance. Okay. So, this tool can give us an estimate of isoform abundance, because it utilizes this maximum likelihood estimates, right, using these expectation maximization algorithms. Okay. So, if you look into the algorithm, you will see like there are variables that cannot be directly measured or quantified, but they can be predicted from the data that is there, and this EM algorithm actually tries to optimize, right, the parameter values so that it feeds the data, right. So, that is an idea behind maximum likelihood estimate, right. Given the data, what is the most likely value of parameters or most likely value of this abundance in this case. Okay. So, this is how it can actually generate these estimates of abundance, and you can also see it can handle how these multiple mappings. Okay. So, now what we are going to do is we are going to discuss some of the alignment free methods that are out there. Okay. So, what we have discussed so far, most of these methods they require some sort of alignment, right, against a reference transcript or a reference genome for quantification. Okay. Now, this mapping step, this alignment step is the slowest step in the full process, right. So, if you look at the pipeline, this is a very slow process, because you have to align this huge number of reads, millions and billions of reads, right. So, that is why these very efficient algorithms were designed, right, the BWT etc., also the HiSET too, right. Now, if we can completely skip this alignment step, and if we can still get to the abundance, right, directly from the read data somehow, okay, and this is what these alignment free methods actually do. Okay. And there are several methods that we will discuss very briefly again, without going into the full statistics, the algorithms etc. So, these tools are called Callisto, Salmon and Shellfish. So, these are the tools we will discuss. There are other tools as well out there. So, let us discuss the first tool, Callisto. Okay. So, it quantifies abundance of transcripts, right. This is the task, right, that this tool does, but it does so based on something called pseudo alignment. Okay. There is no real alignment, actual alignment. So, that is why it is called pseudo alignment or alignment free approach. Okay. And the advantage is this is much faster than the alignment based approaches. Okay. So, you do not have to do this alignment or mapping. So, it is actually much faster. In addition, it is less sensitive to sequencing errors. Okay. So, because you are not aligning exactly, you do not have to worry about these mismatches in the read data etc. So, this is actually less sensitive to sequencing errors that you can get. Okay. So, what is the pseudo alignment approach? Okay. So, pseudo alignment approach is that it actually tries to identify transcripts, right, from which a read could have come from, right. So, given a read, you want to identify which transcripts this read could come from, right. That will serve the purpose for quantification of abundance. Okay. And again, it based on a deep Bruin graph approach for transcriptome. We are not going into the exact algorithm and details, right, but this deep Bruin graph approach we have mentioned also in the transcriptome assembly, and it will come again when we talk about genome assembly. Okay. So, if you are interested in the full algorithm and the details, here is the reference, and also the tool you can download from this link. Okay. That is given here. So, feel free to look into the paper and also the tool and see the options that are there. Okay. Then we have another method which is called Shellfish. Okay. So, again this is an alignment free method, and it can also help in isoform quantification. Okay. So, as I mentioned, this is an alignment free method. So, what it does, it actually utilizes something called an indexing of transcripts. Okay. So, it is very similar idea which we have talked about earlier, is the seeding etc. Right. It creates indexes of certain length, right, k mars of certain k value. Okay. And then it looks for these k mars, right, from the read data. So, it is a kind of like an hash based method. So, it searches for these indexes from the read data against the reference transcript. Okay. And from that it can quantify the transcript abundance, and similar it can actually generate what are the k mars from isoforms, different isoforms, and it can find whether these k-mers are present in the read data, etc. Okay. So, this method can also account for different biases in RNA-seq data. Okay. So, we will talk about these biases in much more detail in the next class, and you will see, right, this model this method can actually model those biases when processing RNA sequencing data. The other tool that is very popular is Salmon. Okay. So, again we will not go into the statistics, I will just very briefly mention what it does. So, it actually uses something called the dual phase statistical inference procedure, and it can also take into account of the RNA sequencing biases, for example, sequence specific biases or GC content biases. Do not worry about these biases, which we will discuss in much more detail. So, there are two phases in this algorithm, as you can see, right. So, one is the fast mapping model that actually estimates expression levels and model parameters. So, it's a model, and it is a model based estimation of the abundance, and then for after the estimation it kind of refines these expression estimates. Okay. So, using these two steps. Okay. So, here is the reference for Salmon, and you can have a look into the original paper, and also you have the tool available for download from this link, and you can explore and learn a lot more about these methods. So, here are the references that we have used for this class. So, what we have discussed is that how we actually go from raw read data to raw count data, right. So, this is the quantification step in RNA sequencing, and this is a very important step, right. So, we have now the abundance of transcripts, right, quantified in terms of numbers, as you have seen, right. So, we have done the hands on, and we have seen, right, we have these abundance values generated from the map data. Okay. And we have talked about some of the tools that can do this very simply, right. We have this HT-sec count and the feature counts. We have not used feature counts, but we have used HT-sec count in our analysis, and we have generated the raw count data. Okay. And this raw count data serves as a proxy for the expression level. Of course, you cannot take this count data immediately as the expression level, because there are a lot of biases in the data that we need to account for before we can actually compare expression levels. Okay. So, we will talk about these biases in the next class, okay, and you will understand, right, how we actually go from the raw count data to the expression level, actual expression level, right. So, that is the idea, right. We want to measure expression level of genes from the read data that we have got. So, we have proceeded a bit, right. We have now got the raw count data, and we can generate that, and from this one we now want to go to the actual expression levels of these genes, right. So, we need to account for a lot of biases along the way. We have also talked about all algorithms that can account for these multiple mappings of reads, right. So, this is a problem with simple tools, right, to HT-sec count or the other tool that we have just discussed, is that they cannot decide, right, whether this comes from a specific isoform or is just count as a gene, right. So, in those cases this is something that can be a problem, right, in where the isoform expression is of interest in your experiment, right. So, this is something that some of the tools can deal with. For example, the RSEM, we have discussed this, and it can provide gene and isoform level abundance estimates. It utilizes something called expectation maximization algorithm. Again, without going into a lot of details, it fits actually a model and try to generate this maximum likelihood estimate from the data. There are other tools that kind of do very similar work. For example, we have something called ISO-EM that also can give you isoform level abundance estimates. We then talked about some of the alignment free approaches, right. So, for transcript quantification, we talked about specifically 3 tools, just mentioned them. So, one is Callisto, the other one is Sailfish, and the third one is Salmon, right. And these tools, they do not require any alignment, right. So, they can go from the read data to the quantification part, right. So, there is no mapping or assembly step. So, this is actually jumping through the mapping part, right. So, again this kind of reduces time requirement, because mapping is the most time consuming process. If you imagine you have to map let us say billions of reads. The other advantage is that you do not have to worry about sequencing errors too much, because these are less sensitive to those type of errors. And these methods are substantially faster than mapping approaches. So, that is why they are quite popular now. So, you can look into those references, and you can see, right, there are time comparisons between these different tools in those papers, and you can see these algorithms Callisto, Selfish and Salmon, they are significantly faster than all those tools that we have discussed that rely on alignment. And some of these models can also incorporate model for biases, right. So, we have a lot of biases in RNA sequencing data, and some of these tools, these alignment free methods, they can also take those biases into account when quantifying this transcript abundance. Okay. So, we will talk about these biases in much more detail, and as well as the methods to deal with these biases in the next class. Thank you.